Hi everyone, my name is Tess Eidem, and I'm a microbiologist and food safety professional in the cannabis industry. And I've been working in cannabis for about close to 4 years now. I got my start in manufacturing, so I was boots on the ground, doing all the extraction, processing, purification, started doing more of the quality aspects, and moved over to consulting with my company, Rogue Micro. I also work in a research partnership with CU Boulder's Environmental Engineering Department in their aerobiology and disinfection laboratory. And today we're going to talk about the microbial risks to cannabis users. And we're going to take a really broad view and cover these three major points today. One, we're going to review the cannabis microbial risks to consumers, both those acute risks, so those are the risks that are going to have some immediate outcome, usually as a result of viable bacterial and fungal pathogens. And pathogen is a specific word that means those microorganisms that can cause disease. We're also going to talk about the chronic risks associated with some of these microbial contaminants, and that is more linked to these non-living harmful biological agents that we'll cover in the next few slides. We're also going to talk about the application of kill steps and microbial reduction technologies in parallel food industries and compare and contrast how food takes preventive controls versus how the cannabis industry essentially sort of abuses these microbial reduction technologies to recover adulterated product. And finally, we'll review recommendations for herbal products from health agencies and professional organizations. So right off the bat, when we look at acute risks versus chronic risks, those acute risks, they require viable microbes, and that means living microorganisms. Those microbes, following combustion or vaporization of plant products, can carry into the smoke and make their way into your lungs. So microbes survive smoking and vaporization. And the chronic risks associated with these microbial contaminants involve their biological agents. So that includes their toxins, their allergens, and immune modulators. And living organisms can elicit these chronic risks, but also non-living microbial components can activate some of these immune responses. And so it doesn't require those microorganisms to be alive or to be viable on the final product. And both these living microbes and non-living biological agents are associated with acute and chronic diseases that we find manifesting in cannabis consumers as well as tobacco users. And what's really important when we're thinking about the risks to cannabis smokers and vaporizers, really focusing in on the flower-based products, is that when we smoke those products or vaporize them, we're actually creating bioaerosols. And these pose really unique risks to smokers that are going to be a little bit different than food safety, but we can use food safety measures as kind of a milestone to start building in these good practices in the cannabis space. So zooming in on these acute risks, again, these require living microorganisms. And we know that viable microbes can survive combustion. We've known that on tobacco products since the 1960s, which you can see cited here. There's plenty of citations here. If anybody has any questions, feel free to reach out, and I can get you some copies of these papers if you don't have access to them. And so we've known that these viable microorganisms can survive combustion, and we can see that, even in this more recent paper from 2022, this research group rigged this cool little aseptic polling technique to take in metered amounts of the smoke, identify those microorganisms by sequencing, and then plate them to see how viable those microorganisms were. And so, we've been doing that with tobacco. There's also a more recent publication that came out in 2023 showing that cannabis vaporization also does not kill microbes, and you can find viable microorganisms in those smoke products that are generated during combustion and vaporization. We also know that based on case studies that Pseudomonas aeruginosa, which is a bacterial species, Cryptococcus neoformans, Rhizopus and Aspergillus, those last three being fungal opportunistic pathogens, are all associated with cannabis use and infections. So cannabis is still a Schedule I drug. It makes it very challenging for researchers, both on the medical side and on the scientific laboratory side, to really thoroughly study and understand some of these risks. So we have to look to case studies, and we have to look to this other paper that came out back in, let's see, which one was this one? It was this 3.5 times. So cannabis users are 3.5 times more likely to have fungal infections than non-users, and that was this 2020 Benedict paper that showed by looking at a bunch of insurance claims, we can kind of gauge some of these risks that cannabis users tend to be more prone to experiencing, and that's fungal infections. And out of those, about 43% of those infections were attributed to aspergillosis. Up here on the right, you can see a cross-section of a cannabis user who had aspergillosis. That's Aspergillus infection of the lung. And you can see some of the nodules that are formed in there, as well as emphysema, which is pointed out by this red arrow here. And we'll talk about emphysema on the next slide. So it's been pretty well understood and established that these microbial contaminants in the plant matter can be vaporized, combusted into these aerosols. Those aerosols then can be collected and plated, and those microorganisms are alive. And we see an increased risk of some of these fungal infections with cannabis users. On top of those acute infections and finding those pathogens in smoke, we can also identify some of these biological agents that microbes produce. So microbes don't have to be alive to be harmful. They produce all sorts of metabolites like these mycotoxins here. So you can see this is a phylogenetic tree. That's just a little tree of life, kind of separating out different fungal species from one another and the different mycotoxins that they produce, sort of binning them together in their relatedness. And there are hundreds and hundreds of identified fungal toxins that have been identified, and we only test for a handful of them. So we test for a couple of different aflatoxins, and we test for ochratoxin. Those are predominantly produced by Aspergillus species and Penicillium species. We do not test for, hundreds of other mycotoxins that could be on the product. And there is evidence that those mycotoxins from other species are present on cannabis product. So it's very important to think about these toxins and that we don't have the full picture. There's also other parts of microbes, these microbial byproducts like endotoxin or LPS, lipopolysaccharide, exotoxins, ergosterol, peptidoglycan, and other components. These are kind of the structural elements of different microorganisms. It's their byproducts, and they also can elicit all sorts of immune responses. Your immune system has evolved to recognize these microorganisms. And here in this diagram by Yang in 2020 shows some of these endotoxins or LPS and allergens, which I'll talk about in a second, interacting with lung cells. And when they interact with these cells, they elicit all sorts of immune activation responses. So you can see all these chemokines and cytokines, basically recruiting more immune cells and interacting with each other, eliciting different maturation of immune cells, again, more chemokines, cytokines being released. And in the end, you can get negative health consequences like asthma, COPD, and lung cancer. So it's very well established that these microbial byproducts can elicit these immune responses, including ergosterol, which is a component of fungal membranes, and it can induce a type of programmed cell death called pyroptosis, which is a result of inflammation. So cells will kill themselves when they interact with and when these immune responses, this cascade of events, create this programmed cell death called pyroptosis. In addition to the toxins, the microbial byproducts or these biological agents, we also have fungal and bacterial allergens. And these work by basically activating another branch of the immune system, very similar to if you have a bee sting allergy or a peanut allergy. You can also develop allergies to fungal allergens. So this is a microscopic view of Aspergillus here. This is one of the microorganisms that I study. And Aspergillus has all these different types of allergens that it produces. And these allergens are recognized by your immune system and elicit a pretty immediate response. And if you have ever gone into a trim room where people are trimming, the trimmers are trimming off mold from flour, or you've gone into a moldy, dusky hotel room and your eyes started watering and your nose started running, you started sneezing, coughing, those are pretty mild allergic reactions, but you can also have much more intense allergic reactions, including anaphylactic shock. So microorganisms do not have to be alive in order to cause harm. And that's what we see in some of these other studies in tobacco and diseases that we see in cannabis users. So these biological agents can also be found in smoke. So not only are we finding living organisms in smoke, but we're also finding all their bits and pieces that can carry over from heavily contaminated product into that smoke. So biological agents like peptidoglycan, LPS, and ergosterol have all been identified in tobacco smoke, and they're associated with these chronic inflammatory diseases like chronic obstructive pulmonary disease, COPD, which is pictured on the right here, and we'll talk about shortly, asthma, bronchitis, and alveolar hypersensitivity. In addition, kind of linking those things together is that smoking cannabis is also associated with more outpatient medical visits for respiratory issues and higher rates of pulmonary diseases, including chronic bronchitis and emphysema, which are both types of COPD. So here we have COPD and a healthy lung. If you zoom in on a part of that lung here and you look at the little branches that are carrying air, these branches are called bronchioles. And they carry air into these little sacs where air exchange occurs, and those little components are called alveoli. So in a healthy lung, air exchange goes through these tubes and happens here in the alveoli. In emphysema patients, you find that the alveoli are damaged. They're collapsed. There's a lot of inflammation and damage going on in those regions of the lung. And in chronic bronchitis patients, you see a lot of mucus in these little branches and inflammation, really preventing the air from getting to those alveoli and allowing for efficient air exchange to occur. So it's thought that these components really play a big role in tobacco users in eliciting this immune response, and it's really linked to these diseases. And there's more and more evidence that these more chronic effects that we see in cannabis smokers could be linked to heavy bioburdens on the cannabis product. Even when treated with remediation technologies, those skeletons of those microorganisms are still present on the product, and they still pose a risk to cannabis users, specifically smokers and folks who vaporize. So as manufacturers, cannabis manufacturers and cultivators should really be considering these risks, because that's what you have to do in food safety. So food manufacturers have to implement different preventive controls. They have a whole food safety program in addition to their GMPs and prerequisite programs that really hone in on the risks that are biological, which we're talking about here, but also chemical and physical risks. And they have to put in preventive controls to make sure that they're not just covering stuff up at the end, but that along their entire process, they are controlling and managing all of these different risks. In specific food processing techniques, there are preventive controls called process preventive controls. Now these include kill steps and microbial reduction steps to address risks identified in your food safety plan. So the first thing that you do is build a hazard analysis or a HACCP plan, and then you build in all this supplemental material to basically proactively address any risks that you identify in your HACCP plan. And these kill steps and microbial reduction steps are used, again, preventively. They're not used after you fail. They're used preventively on product in line with your food safety plan, prerequisite GMP, sanitation program, employee hygiene practices, pest control, good hygiene practices, all of that has to be done. These process preventive controls must be validated within 90 days. You have to show with data, validated data, that there's a scientific basis that this process preventive control is actually controlling the hazard that you identified in your hazard analysis associated with a specific product and process, and establish the critical limit. So that's the time, the temperature, the concentration of ozone, the dose of irradiation, etc. And you can see in this pyramid here kind of this all of the stuff that you have to do with these process preventive controls to really monitor, are you getting to the right temperature for the right amount of time? Have you calibrated your thermometers? How are you measuring it? How are you verifying that the people doing the monitoring are, doing it correctly? Are you going back and checking batch records? Are you making sure that there's biannual training? How are you verifying that you're monitoring this appropriately? And then validation. In the food safety world, validation is very specific. It means you have a preponderance of scientific evidence in studies led by a PCQI, such as myself, that this method is actually controlling the risks that you've identified. And these process preventive controls must be reproducible. You should get the same result every time, monitored by your operators, verified regularly, and audited annually. And in cannabis, there's no such thing as a cannabis kill step. There's been no validation for flower products in the cannabis industry. And a kill step in the food world has a very, very specific meaning. A kill step must get a 5 log reduction in most food products to be called a kill step. If it gets less than a 5 log reduction, which I'll talk about in a second, it's called a microbial reduction step. So kill step, again, needs a 5 log reduction. In food safety, a 1 log reduction is 90% reduction, 2 log is 99. So a 5 log reduction would be a 99.999% kill rate. And right now there is no validated method in the cannabis space on flower products that are validated to get you a 5 log reduction. So there's no such thing as a cannabis kill step. In addition, these technologies are not used in line with good practices. So you find that they're not used preventively on high quality product, to protect shelf life or protect an end consumer. They're used reactionarily to recover adulterated product that has failed microbial testing. And that process of recovering failed product is called remediation. It is not allowed by FDA food safety standards or FSMA, Food Safety Modernization Act. You cannot recover adulterated product in this way with these methods. And so it really goes against all FDA guidance in parallel industries as far as food goes to be using these technologies the way that they're being used. Even if they're claiming that they're being used as a kill step preventively, it's usually because the cultivator is having, micro challenges in their garden and they can't pass without this technology. So they just use it, on every single product, every single batch that comes out. And that's not actually preventive controls. That is, again, it's remediation. It's recovering adulterated product, which again, you cannot do according to FDA FSMA rules. So since we don't have FDA in cannabis, we don't have any FDA oversight or any of these food safety plans or product safety plans that we have to follow. We don't have preventive controls that we have to put in place in cannabis. What are other agencies saying about similar herbal medicinal products and culinary medicinal products? Well, they say we shouldn't do this either. So when you look at like culinary herbal products, Western Growers Association, and I quote, say that these safety programs should focus on preventing adulteration by microbial contamination because these herbs are often eaten raw and without a kill step. And they say to do that to reduce or eliminate human pathogen levels. So you want to make sure that in your good practices, you have a clean environment, You have, you're really focusing on contamination prevention, which is also outlined in this diagram from the 2022 Bedrocan document, The Good Medicinal Cannabis Cultivation Practice, which shows that you really need to build this entire ecosystem of prevention, cleaning, personal hygiene, air filtration, make sure that you have a good HVAC and getting right number of air circulations per hour, pest prevention, and making sure that you are cleaning up dead and dying plants. If you've got infection, you need to quarantine those plants. So there's a lot of good practices that really focus on contamination prevention, and that there are other agencies that have explicitly spoken out against the abuse of these remediation technologies like ozone and irradiation. Specifically, the United States Pharmacopoeia, which came out with this document, Cannabis and Fluorescence for Medical Purposes, USP Considerations for Quality Attributes. They specifically say, and I quote, treatment methods such as irradiation should not be used as a means to remediate cannabis contaminated above the allowed limits. That is how these technologies are being used in cannabis. When you fail, you remediate, and then you recover and test that product into compliance, which you cannot do in food, and it's explicitly forbidden by the United States Pharmacopoeia in their recommendations. The European Medicines Agency and the World Health Organization also speak out against the abuse of these technologies, basically saying that microbial reduction treatments cannot replace good agricultural and collection practices, that's what GACP stands for, or GMP, good manufacturing practices, and or disguise products that are contaminated or unfit for consumption. And they also say if you are going to use these treatments in line with good practices, because ozone and irradiation and other types of microbial reduction technologies can be used responsibly, and they are in other industries, if they are being used on compliant product with low bioburdens without detected pathogens, you've got to label that product so that your consumer knows it's undergone some kind of treatment. And that's what the World Health Organization says as well, that these good practices need to be followed, and treated products, and they specifically name irradiation, must be labeled. And again, these organizations are coming out against the abuse of these technologies to recover failed product. So we don't have FDA oversight, but we do have recommendations from all these organizations and parallel industries that we can pull from. So in summary, smoking cannabis has both living and non-living microbial risks associated with acute and chronic pulmonary diseases. And all hazards need to be considered when we're doing these risk assessments, if we're doing them as healthcare providers, if we're doing them as regulatory bodies, if we're doing them as manufacturers. These are all risks posed to the consumer that we must consider when we are evaluating and putting in our preventive controls. These cannabis microbial reduction technologies like ozone and irradiation have not been scientifically validated or routinely verified. Critical limits have not been established to reduce viable pathogens. We don't have the treatment time and the radiation or the ppm of ozone locked in for every single experiment. We do not have those critical limits established. They have not been published. They're not anywhere. You can't find them. And so you don't even know as an operator if you're going to treat for a certain amount of time with a certain concentration of something. Sometimes you won't even know what concentration you're using or the dose you're using because it's proprietary. But those are part of your critical limits, and the operator needs to know that they're hitting those critical limits. I don't cook a hamburger, and when someone asks me how hot the internal temperature was, I say it's proprietary. No, I've got to know that I'm cooking it to a certain temperature for a certain amount of time. In addition, these microbial reduction technologies do not remove non-living microbial agents on cannabis. So it leaves behind all these skeletons all over the product. So if you have really heavily bioburdened product, with a high TYM count, total yeast and mold, high coliform count, those are intestinal gram-negative bacteria, high Enterobacteriaceae, high aerobic bacteria counts. Those are all threshold levels, right? And you get above that threshold and your product fails. But if you bring them down to get them compliant and pass third-party testing, which again, you can't do in any other industry except for cannabis, then you're leaving all these skeletons all over the product with all their allergens, all their microbial components, and those can still be immunoactive. They can still lead potentially to some of these chronic diseases that we see as a result of exposure to these microbial agents. And again, these microbial reduction technologies are used to recover failed product. This is against all food safety recommendations. Even the FDA has oversight over dog food production, and you're not allowed to do this with dog food, but most states allow it for cannabis production. So it's poor practice, it's not allowed, and all of these other agencies are speaking out against it. So the best approach is really to cultivate with good practices to produce pathogen-free, low bioburden cannabis product. And this is really intended to protect consumers from acute and chronic diseases. And there are agencies out there who are trying to build good practices around cannabis cultivation and processing and packaging. I know ASTM, Americans for Safe Access, AFDO, ASTM, they're all very dedicated to building a culture of safety. So I'm hopeful that with buy-in from some of these manufacturers and really seeing that it does pay off in the long run to be preventive. We know this from the food industry, that it's much more beneficial for a manufacturer to preemptively address their contamination issues at the root cause than to try to recover adulterated product. And so I'm very hopeful that the cannabis industry can step up and meet at least basic food safety guidelines. You know, if we can do it with dog food, we can do it with cannabis. And if anyone enjoys this presentation or has any other questions, I'm doing more seminars like this. I would like your feedback. And if you want any of these publications, I have close to 40 citations in this presentation. Please reach out. I want to make sure that we are educating the cultivator, the manufacturer, the regulatory bodies, and the consumers, because consumers don't know that they're smoking product that failed and was recovered. Most of the time, these products are not required to be labeled, and there's no way to ask your bud tender. And so you think you're going and getting something safe, but these technologies like ozone and irradiation are not necessarily making products safer. In fact, I would argue that they are allowing potentially unsafe product to pass compliance testing and get into the hands of consumers. And that's not the way that we want this industry to go. I hope you guys all enjoyed this presentation, and please give me your feedback. I look forward to hearing from you.